Hi, good day everyone. Welcome to FST 20203 Animal Breeding class. I'm Associate Professor of Technology Dr. Hasnita binti Chair Harun will deliver the topic of Mendelian genetics for the week 2. So before we go further, I want to a little bit on the definition of the genetic. So it is referring to the science of heredity. So genetics is the branch of biology that deal with heredity and also contribute significantly to the variation of organisms. Genetics explain how traits are passed from one parent to the offspring to the next generation, and the information um, that relies in this uh, trait is the reason why individual within a species is differ significantly from one to another. And at the molecular level, chromosomes carry hereditary information in the form of genes, which are specific sequences of DNA nucleotide, ATCG, and this nucleotide um, is later being translated to code for the protein and being expressed to produce the phenotype, the one that we can observe, physically observe. So if you could see the flow of the genetic information, start with DNA, where the genetic blueprint is stored in um, chromosomes. And from this chromosome, the genetic information will be transcribed through messenger RNA molecules from DNA to RNA, and then later it's being translated through functional molecules that determines trait, and this is at the protein level where it is code for the protein and it will express the uh, that particular trait. So this is what we call these three stages of genetic information flow. We call it the central dogma of molecular biology that shows how genetic information flows from DNA to RNA and to protein, and ultimately it will determine an organism characteristic. I hope you could search more information about central dogma, and you have to understand how DNA is transcribed to the RNA and how RNA is later being translated to protein. So you have to look, search for the process that occurs, like DNA translation, DNA transcription, and so on. So I hope you could find some time to search all this information for you to further understand the process that happen, that explain how the genetic information is flow start from DNA to RNA and transcribe translated into protein before it could be expressed to produce when it expressed, the trait is expressed, that is the one that you could see physically, such as your eye color, your hair color, and so on. So chromosomes occur in pairs within cells. So you have to understand that each of these chromosomes contains lots and lots of genes that position in this chromosome, and this pair of chromosomes are called homolog chromosomes. So chromosome always comes in pair. That's why we call it homolog chromosome. So homolog chromosome refers to the one copy that the chromosome comes from your parents, each of your parents. So one from your mom and one from your dad. So each of these chromosomes will combine or attach or pack together to produce homolog chromosome. So this homolog chromosome, they carry genes for the same traits but may have different version, what we call allele. So allele is the different version of uh, gene for that similar trait uh, of those genes. So each parent will contribute one chromosome of each pair during sexual reproduction. So this will create new combination of genetic information in offspring through fertilization. So this figure B11 here showing the homolog chromosome. So homolog region code for the same gene. So homolog chromosome contains DNA that codes for the same gene. So in this example, both chromosomes have all the same gene in the same location, represented by the strip color here, but with different version of those genes, because one of these comes from the mother and another one on the right side here comes from the father. So these two chromosomes pack together and we call it homolog chromosome. The handsome man here is Gregor Johann Mendel. So you cannot study heredity without knowing Gregor Mendel, as he was coined as the father of the genetic. He was born in 1822 and he studied botany and learned the scientific method of crossing the pea plant, and he conducted thousands of control courses of the pea plant, and he come up with three uh, what we call three Mendel's law. So we will learn about that later. So before that, um, we have to understand the theory of the Mendel, what he contribute to the heredity and the genetic. Now, actually, Gregor Mendel challenged the blending theory. So before that, heredity was regarded as a blending process where we believe at that time they believed their offspring characteristic were essentially a dilution of different parental traits, like when you mix between paint colors, they combine together and then you could see the color of combination, a mixing of that different colors. But then Gregor Mendel challenged that blending theory. He come up with this new discovery through careful experimentation. Mendel, what he discovered is that he could demonstrate that inheritance involves discrete unique we call gene that maintain their identity across generation rather than blending together as explained by the theory before Mendel. Mendel chose a garden pea plant to study heredity because they were easy to cultivate, short generation time, produce many offspring, and also distinct observable trait. And most importantly, he could control their breeding by manually transferring pollen between the flowers. So here is the 7 traits that Mendel studied. So he um, examined 7 distinct characteristics of pea plant. The first one is the seed shape, round or wrinkle. The second one is the seed color, yellow or green. Flower color, purple or white. Pod shape, pod color, flower position, and also stem length, either tall or dwarf. So these are 7 traits that Mendel observed in the pea plant. So from his discovery, Mendel's predicted the first one is the concept of gene, where hereditary factors determine trait. The second one is gene occurs in pair. So what he understood that each organism carries 2 copies of each gene. And the third one is gametes carry 1 gene. So each of the gamete, either sperm or egg, it contains only 1 gene from each pair. Before we go further, it is essential for you to understand um, several genetic terminology that is very crucial for you to understand, and that because this terminology will be used frequently in this subject matter. So the first one is gene. So gene is a unit of heredity, a section of DNA sequence that encode a single protein. So gene relies within the genome. So means that genome is very, very big because genome represent the entire set of gene in an organism. So the whole gene that an individual and organism could have. So that is genome. And allele refers to 2 or more versions of the same gene, like different flavors of a trait. You have like for all different color of a trait. For instance, you have black hair, you have brown hair, you have blonde hair. So they are alleles that represent 2 or more version of the same gene. So you have 1 gene, so it is expressed different ways. So that is allele. So that is the allele occupy the same position on homolog chromosome. And next is locus. So locus is a fixed location on a strand of DNA where a gene or one of its allele is located. So that is locus, and more than one locus, we call it loci. Next one is trait. So trait is a characteristic determined by a gene or genes. So means that trait is the things that you can observe, you can see physically. For instance, hair color, whether it's black, brown, eye color, whether it's blue, green, skin color. So all those things that uh, you can see, what we call the phenotype. So it is a characteristic determined by a gene or group several more than one genes. So that is trait. Homozygous is having identical allele from one from each parent for a particular trait. So compared to heterozygous, when you have two different but similar allele for a particular trait. So that is the differences between homozygous and heterozygous. Homozygous when you have identical allele, heterozygous when you have two different allele for a particular trait. So here is the comparison between dominant and recessive allele. So dominant allele refers to the allele of a gene that masks or suppress the expression of an alternative allele. So the recessive allele. The trait appears in the heterozygous condition written with a capital letter. So the example that I put here is a capital letter T. And compared to recessive allele, it is refers to the allele that is masked by dominant allele. The trait does not appear in the heterozygous condition. It is being masked. And it is only appear when it is in the state of homozygous recessive. So recessive allele is written with a lowercase letter. I put here example of lowercase letter t. Here I put example of stem length in piece, long and short stems in piece. And I put T as the allele for capital letter T for dominant allele, and that encode for tall, and small letter T for dwarf, that represent the recessive allele. If you look at the genotype, you have three types of genotype, different genotype, which is homozygous tall, represent by both capital letter T and both small letter t for the homozygous dwarf plant, and another genotype for the heterozygous tall plant. So the trait it is for the stem length. Tall plants grow significantly taller than dwarf plants. So the trait will express that you could see either the plant is tall or the plant is dwarf. So this is you can like predict back the genotype for the tall plant, and what is the genotype for the dwarf plant, and from there you can predict allele. You can assume allele that represent each of the gamete for that particular genotype. So genotype versus phenotype. The genotype is the genetic makeup of an organism. So it is the actual allele present in its DNA form. So this is the genetic code that may not always be visible. You cannot see this code, but then it is relying inside your body that code for a particular gene or genes, so that it could express the trait, where you can see the phenotype. So basically the phenotype is the physical appearance of an organism, the one that you could observe it is observable trait resulting from the genotype and also combined with the effect of environmental factor. So you could see either the plant is tall or dwarf. So that is the phenotype for the stem length. Causes and generation. For the monohybrid causes, it is a genetic cause involving a single pair of gene, only one trait because of single pair of gene, and parents differ by a single character. So for this monohybrid, the first generation is the parental generation. So this is the original two individuals being crossed together, and this mating or cross will produce F1 generation. So this is the first fetal generation. Offspring from a genetic cause of the P generation, and later it will produce F2 generation. For the next generation is F2 generation. This is the offspring that being produced when the F1 individuals are crossed together. So you could see in this slide from Mendel's breeding generation, it starts with parent, P generation, so produce F1 generation, and then F2 generation, and then F3 and beyond. So we'll continue to F6, F7, and so on. So again, I show you the 7 characteristics in face because I want to take example from this characteristic in face to show you the monohybrid and dihybrid crossing in pea plants for you to further understand how monohybrid cross happen and how dihybrid cross happen. So for the monohybrid cross, it is a cross that examines the inheritance of a single trait. So we use the example of stem length in pea plant. So the cross is homozygous tall mate or cross with homozygous dwarf. So both parents are true breeding because both of it are in the form of homozygous. So they consistently produce offspring with the same trait when self-pollinated. So you have to look at the notation that I put here. Capital T for allele for tall, small letter, lowercase letter t represent the allele for dwarf, and capital letter is for dominant effect, and lowercase letter for the recessive effect. So now we want to further understand about the monohybrid cross. So cross of parents differ by single trait only. That's why it is monohybrid. So crossing two pea plants that differ in stem size. For this example, we use stem size. It is either tall or short or dwarf. So I put here the allele represent as capital letter T and lowercase letter t for dwarf, capital letter T for tall. And now we predict the homozygous tall plant represented by these two alleles, and homozygous dwarf plant represented by the small letter, both of it small letter t. So we cross it together between the long and short stem. So this is monohybrid cross-post-strand length. So P parental through breeding homozygous plant, either tall, we cross between tall homozygous dominant and dwarf plant for the heterozygous. And we will produce the F1 generation, which is all of them is heterozygous. So the F1 generation is um, heterozygous. The genotype is all tall plant, and the genotype is all capital letter T and lowercase letter t, and the phenotype is all tall plant. So F1 result is all tall parent cross when we cross homozygous tall with homozygous dwarf plant, all F1 offspring are tall. This happens because each parent can only contribute one type of allele. So tall plants give capital letter T, dwarf plant give lowercase letter t, resulting in heterozygous offspring. Since capital letter T is dominant, so we could see that all heterozygous plant is appear tall, even though they carry the recessive allele, because we know that the recessive allele is always being masked, being filtered by the dominant. Okay, now we have the Punnett square or P square. So it is a powerful genetic tool. So it is a diagram that predicts the genotype and phenotype of offspring from a genetic cross. Punnett square was developed by British geneticist Reginald Punnett in the early 90s. From this Punnett square for a monohybrid cross, we could use 2 times 2 grid that looks like a window pane with 4 boxes. Looks like this. So for you to use a Punnett square, the first step is you have to determine the genotype of the parent organism that you're working with. And next you have to write down your cross, your mating. And let's say you draw a P square. Now you have parent genotype that you predict. And then you already have your cross. Now you can split the letters of the genotype for each parent and put them outside the P square. Next is you determine the possible genotype of the offspring by filling in the P square. And lastly, for each of the Punnett square that you created, you have to explain and summarize the result for both genotype and phenotype of the offspring produced here. For this example, the crossing between homozygous cross cross will produce genotype that is 100% heterozygous and with phenotype of 100% tall plant. For the F2 generation, so just assume that we still continue from F1 generation. So we assume that F1 generation is self-fertilized. So the next monohybrid cross would be the crossing between heterozygous cross. So from this cross will produce F2 genotype that is one uh, homozygous dominant tau, two heterozygous tau, and one homozygous recessive with dwarf. So the genotypic ratio is 1 to 1. For the F2 phenotype, you could see only two types of the phenotype is produced, which is three of them are tall and only one is dwarf. So the phenotypic ratio is 3 tall and 1. So the secret of Punnett square is so simple. The key to the Punnett square is that you have to determine the gamete of each parent by splitting the genotypes of each parent. So if this is your cross, then you have to split the gametes, split the genotypes so that you could produce the gametes from each of the genotype or the parents that you want to. Now you already have the gamete, so you can cross between gamete for parent 1 and gamete for parent 2, and you could predict the offspring that the possibility of you to have offspring for that particular crossing. So what you should understand is that the 3 to 1 ratio is a hallmark of monohybrid crosses between heterozygous parents. For every 4 offspring, we expect approximately 3 to show the dominant trait and only 1 to show the recessive trait. So this ratio provided crucial evidence for Mendel's principle of segregation. I also put another example here using the flower color. Uh, the flower color is purple for dominant and white for the recessive. And similar, when you cross between homozygous purple with a homozygous white, of course you will have heterozygous purple for F1 generation. And later, when you cross the F1 generation, the heterozygous purple cross the heterozygous purple. So for the F2 generation, for the genotype ratio, you will get 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive, compared to the F2 generation for the phenotype ratio. Similar also with the stem length, you will have the ratio of 3 to 1, 3 purple and 1 white for the recessive. So here what Mendel's want to describe is the Mendel's principle of dominance. And the second one is principle of segregation. So principle of dominance is when one allele can mask the expression of another allele. So you could see that the dominant allele is expressed in the F1 generation while the recessive allele is being masked or hidden. And for the second principle, which is principle of segregation, when gametes are formed, pairs of hereditary factors, the allele become separated so that each gamete will receive only one allele from each pair. I put here also the example of cystic fibrosis, CF. You can go through this slide and do some exercise on this. Here is another example of Gaucher disease. You can you can go through and try to understand the inheritance of this disease as well, using the monohybrid cross. Now we want to learn about dihybrid crosses. So compared to monohybrid crosses that involve only a single trait, uh, the differences between monohybrid and dihybrid cross is that dihybrid cross is the crosses that involve parents that differ in two independent traits or characters. So here I give example for the flower color, purple and white, combined because it is dihybrid, so it needs to have another um, characteristic, which is the second one is stem length. So we have two here, flower color, and the second one is stem length. So each of it represent with a letter T and T. So here is the dihybrid cross between flower color and stem length. So possible gametes for parents is homozygous um, dominant for purple and tall, and homozygous cross with the homozygous recessive, which is white, and a dwarf plant. So you can predict the F1 generation that will produce all tall and purple flowers with both of the gamete um, in the form of heterozygous. So similar to monohybrid, for the F2 generation, we assume of self-crossing between the to produce the F2 generation. So Mendel observed four phenotypes, which is tall purple, 9 out of 16, tall white, short purple, and only 1 over 16 is short white. This represents both homozygous recessive for flower color and also the stem length. So this is the gamete formation. So self-cross F2 will produce ratio of 9:3:1, which is 9 tall purple, 3 tall white, 3 short purple, 1 short white. So how you know about this is you have to understand the Punnett square. You have to understand the gamete that you need to put for each of the p square outside the p square, and you have to predict the genotype of the offspring inside the P square. Okay, now we come to the Mendel's third principle, which is principle of independent assortment. So this referring to the members of one gene pair segregate independently from another gene pairs during gamete formation. So this one could be illustrated from dihybrid crossing that you learned just now. So this means that the inheritance of one allele, like the one that we go through just now, the flower color does not influence the inheritance of another trait. In our example, is the stem length. So they segregate independently between flower color and also stem length. They are not independent to each other. So the genes are shuffled independently. So this independent assortment combined with random fertilization, so this creates tremendous genetic variation. One of the key advantages of sexual reproduction over asexual. Thus cross is a cross of an individual with a homozygous recessive individual. So this is particularly useful when you have an individual with an unknown genotype. You want to predict the genotype of that particular individual. So you need to do test cross. So for example, a plant with purple flower can either be homozygous capital letter P or heterozygous. It could be either one because we know that dominant effect will mask the recessive allele. So if you observe purple color, you can't really sure. You cannot tell that the genotype is either homozygous or heterozygous. Uh, be, therefore, you cross the plant with homozygous recessive, white flowers homozygous recessive, so that you can predict back, you can guess back the genotype of that um, uh, individual that you cross with homozygous recessive. So I want you to use this as your class activity. You have to predict the answer for this test cross, and I want you to justify as well why why we need to cross with homozygous recessive only but not heterozygous or homozygous dominant. So please justify your answer. So here is dihybrid test cross. So similar, if you had a tall purple plant, so how would you know the genotype behind it? Because it has four possibilities of this tall purple plant for the genotype, and because of test cross, in order for you to predict the genotype of this individual, you need to cross it with homozygous recessive for both of the trait that you want to study. For both of the stem length, you have to cross it with dwarf and white flower so that you can um, predict the genotype of the other parent that is unknown. So I want you to justify to produce your result in the class activity. I will open a channel in the eCampus for you to drop down your answer with justification. why um, your answer is like that. You have to justify, and also I want you to also justify why for both test course we need to cross it with homozygous recessive individual. So that's all for today. Thank you very much.